Hello, I am Reil, and today I would like to share this pastel painting process with you while talking some mutation nonsense. I am painting a planked walkway from the Lake Baikal. The reference was taken by my high school classmate who followed her childhood dream to study seals and a decade after our graduation worked on a project concerning her favorite animal, the Baikal seal or Pusa sibirica. This painting turned out to be perfectly colorblind friendly, and this prompts me to talk about color vision. I have to take it into account as a researcher when I choose how to visualize the data. The simplest example would be fluorescence microscopy images that are obtained using red, green, and blue fluorophores. So you may be prompted to use a standard green and red scheme, but it would be challenging for those with deuteranomaly, the most common type of red-green color blindness. How does color vision work and how can it fail? We are trichromatic and have three types of cones in the retina, like your computer screen, red, green, and blue. They differ, among other things, by which type of opsin or receptor they express. The short wavelength sensitive or blue, intermediate or green, and long wavelength sensitive or red. Each one is encoded by its own gene, and mutations in each gene can cause problems. Complete loss of OPN1SW function causes tritanopia, and mutations that ruin this protein incompletely cause tritanomaly. These people see the world in red-green colors, but this is a very rare condition. The reason for it is that OPN1SW gene is located on an autosome, not on the sex chromosome, and each one of us has two copies of this gene, one from the mother and one from the father. If one copy is broken, the other one can compensate. And there are not so many broken copies in our population, so the chances of two such people having a child are quite low. Things are different with red-green color blindness. If you have a mutation in the green cone gene, OPN1MW, and you are male, you have a problem. OPN1MW is located on the X chromosome, of which males have only one. So there is no second allele that could compensate, and there is quite a large portion of people who have such mutations and see the world in shades of yellow and blue. Incomplete loss of function is called deuteranomaly, and complete loss of function is called deuteranopia. Mutations in the red opsin gene OPN1LW are quite similar. Like the green opsin gene, the red opsin gene also lies on the X chromosome, and people who have mutations in it see everything in shades of yellow and blue. The difference is that they cannot detect the wavelengths on the red side of the visible spectrum, and thus red looks darker to them. It probably causes more problems than mutation in the medium wavelength opsin and is better selected against, so protanopia and protanomaly are not very common even in males and are exceedingly rare in females. Possibly defective alleles of the green opsin gene linger in the population because they are selected for. There are indications that such people are not fooled by certain types of camouflage. So there might be a silver lining to having deuteranomaly after all. But look at these absorbance spectra. Do you notice how close the medium and the long wavelength peaks are? And they both suspiciously lie on the same chromosome. There must be a reason for that, and indeed there is. The common ancestor of all tetrapods, or four-legged animals, had four opsin genes and had decent color vision. But when reptiles dominated the ecosystem in the Mesozoic, our mammaliform and the mammalian ancestors were nocturnal. And at night you can't properly use color vision as the light intensity is too low to activate the cones. So there was little selection pressure to eliminate mutations in the cone opsins. This way, our ancestors lost two of the four cone opsin genes. Then, our less distant ancestors transitioned to living in daylight and to eating fruit, and suddenly, by evolutionary standards, the ability to discern red and green became very useful. Now, gene duplications occur all the time, but are rarely supported by natural selection, and the second copy accumulates deleterious mutations and is soon lost. But this time it was different. In the early world monkeys, the gene got duplicated and quickly accumulated some useful mutations that moved the spectra of the two opsins apart. In the New World monkeys, this duplication did not occur, but trichromatic vision still provided benefits for survival. So in most New World monkeys, all males have dichromatic vision, having one short wavelength opsin and one long wavelength opsin encoded by an X chromosomal gene, just like in most other mammals. However, this X chromosomal opsin gene has accumulated variation within the population, so females who have two X chromosomes have a chance of having two different alleles of this opsin gene and have trichromatic vision. But isn't it sad that only females are reaping the fruits of trichromacy? Howler monkeys got lucky and evolved trichromacy in both sexes, just like the Old World monkeys independently. Meanwhile, owl monkeys didn't need color vision for their nocturnal lifestyle and lost their blue cone opsin entirely. Now, usually diseases are named after the doctors who first described them, much more rarely after the first patients, but occasionally the doctor and the patient are the same person. One such case is well known. John Dalton was the first to describe congenital red-green color blindness, or deuteranomaly, which he himself had, a condition we know as Daltonism. But there is another lesser-known condition that I'd like to cover today, myotonia congenita, and specifically Thomson syndrome. So let's go back in time to 1815, when Asmus Julius Thomson was born in Schleswig. From a young age, Thomson had a peculiar problem. After any voluntary movement, his muscles would spasm. These spasms could be avoided if he warmed up his muscles first. For example, as a kid, he couldn't just get up quickly from a chair if someone called him suddenly. If he tried to stand up too fast, his legs would freeze in a painless spasm and he couldn't walk. If he pushed it, he'd fall over. But after a bit, his muscles would relax and then he could run as fast as the other kids and was even more resilient. Cold weather made his symptoms worse. Adults didn't understand what was going on with him and even punished him for what they thought was disobedience. Thomson managed to keep his problem hidden, and it didn't stop him from becoming a doctor and having a family. His younger son inherited the disease, and while it's something you can manage in everyday life, it's a big problem for military service. Unfortunately, people with myotonia often look quite athletic because their muscles are hypertrophied, so nobody believed Thomson's son was sick and accused him of dodging the draft. To defend his son, Dr. Thomson wrote a manuscript titled Tonic Spasms in Voluntary Muscles Due to Hereditary Psychic Disposition, Muscular Ataxia. The term ataxia wasn't quite right, but let's cut him some slack. It was 1876. Anyway, Thomson described what he and his family experienced, along with 3 other families of his patients. Later on, this condition was named congenital myotonia. Thomson disease is a type with autosomal dominant inheritance, while another form with recessive inheritance is called Becker disease. Don't confuse this with muscular dystrophy, also named by Peter Emil Becker. Over a century later, in 1994, a team led by Thomas Jentsch figured out the molecular mechanism behind congenital myotonia. They found out that the disease is caused by mutations in the chloride channel gene CLCN1. This channel helps muscles stabilize their membrane potential so that they don't contract without a signal from the nerves. The researchers sequenced the gene in the known living descendants of Dr. Thomson and in several Canadian families with myotonia and identified several mutations. The channel is made of 2 identical CLC1 protein subunits that have a common gating mechanism, and this gives rise to 2 possibilities. If the mutated subunit affects its normal partner, that mutation would have a dominant inheritance pattern and have its effects even when present on just one chromosome. This is what happens in Thomson syndrome. If, however, the defective allele doesn't mess with the normal one, you would need 2 mutated copies of a gene to cause the disease. This type of myotonia is called Becker syndrome and is inherited in an autosomal recessive way. In the 30 years since that study, many more mutations causing both types of congenital myotonia have been discovered. Some patients have similar symptoms but normal CLCN1 gene. They have mutations in the sodium channel gene SCN4A, and this condition is called congenital paramyotonia, just to keep things clear. Oh, and by the way, myotonia isn't just a human condition. The molecular link between myotonia and CLCN1 was first established in a mouse line that had a transposon insertion in this gene. Beyond that, have you ever heard of Tennessee fainting goats? They don't actually faint from surprise or fear, they suffer from myotonia. Loud noises cause their muscles to spasm, making them look as if they're fainting. You can find the videos online, and don't worry, the goats are not feeling any pain. I'll leave you with the goats for now. Please leave a comment and a like or a dislike and subscribe before you go, and I'll see you in the next video.